as you say. So.
Hola, muy buenas tardes a todos. Muchas gracias por estar presente aquí en el seminario de todos los viernes. I will switch to English now. Yeah, so it is my great pleasure to introduce today Albert Guskop. He is a structural biologist. He got his PhD in 2009 in the Free University of Berlin. And he, now in his lab, he combines macromolecular X-ray crystallography, single particle cryo-IM, and modeling in his original investigations of membrane proteins and macromolecular complexes. The main line of his research are structural and functional characterization of solute carrier family 1 of transporters, bacterial metallohomeostasis, protein translation apparatus in pathogenic fungi. Albert's group has significantly contributed into mechanistic understanding of transmembrane transport, and he has over 20 publications during the past 5 years on this topic. Besides these publications, he has more than 50 peer-reviewed papers, and he has received more than 3,800 citations. He has received various personal grants from NWO, including the VD grant. As a recognition to his scientific excellence, Albert was awarded an honorary professorship of Moscow Institute of Physics and Technology in 2019, and a Medal of Excellence for Contribution in Science in 2021. In addition to that, Albert is a member of Program Committee monitoring the educational standards of master programs at FSD, a regular member of PhD committees, and an active peer reviewer for journals such as Science and Nature. He also is a grant reviewer for many national and international funding agencies, including the CNRS in France and the Helmholtz Foundation in Germany. So today he's going to talk to us about his latest research, and we thank you very much for being with us, and welcome.
Thank you very much, Violetta, for such an extended information. So is it on? The mic is on, so you can hear me well. Perfect. So it's my great pleasure to be here. Actually, it's the first time I'm in Mexico. So thank you, ALS, for arranging that in part. So indeed, I'll talk today about not only latest research, but I will give you a broad overview of what we've been doing in this particular topic on solute carrier transporter family 1. It's actually probably about 15 years of research compressed in one talk. So apparently I will not manage to go into details for everything, but feel free to interrupt me at a certain point, because I do understand not all of you are hardcore structural biologists, so some things might be very new for you. So let's just continue. There is a extended also outline of things I will try to cover. I will not go through it right now to save some time, but I will start immediately with the importance of solute carrier family of transporters. So there are plenty of these families. Actually, there are over 52 families now with over 500 individual members, and we are talking now about mammalian transporters, human ones. And if we look at transporters which are linked to it does work to some extent. So if we look at the transporters which are linked to Mendelian diseases, so those which are inherited, so we can see that quite a few actually are involved in the transport of neurotransmitters. So, and I don't have to explain you, right, about the importance of neurotransmitters and their transport and regulation. So you must have heard of transporters which are involved in, let's say, transport of such things like dopamine, noradrenaline, et cetera, But it turns out that one of very important neurotransmitters as well is just an amino acid, glutamate. And for transport of glutamate, there is a specific family, so SLC family 1, which actually includes several members, AATs, so-called excitator amino acid transporters, and neutral amino acid exchangers, which I'm going to cover as well in this talk. And again, you don't really have to, and you probably cannot even from such a distance, read all the diseases which are linked to the malfunction of these transporters, but I will just highlight that if you have troubles with those transporters, you're most likely going to develop some quite significant neurological diseases. And while some of actually also neutral amino acid exchanges are linked to cancer, but we will talk about this a bit later. So here I will jump back actually from human transporters to archaeal transporters, which turn out to be quite a good model system to study mammalian transporters as well. The reason is it's very mundane in a way. It's simply because it's much easier to work with archaeal transporters, given their stability, well, they express well, they can be purified well, et cetera, et cetera. So if we, so now we have to jump back here. So hence, historically, our group and other groups in the world, they actually started to investigate these transporters just using archaeal homologs. And it turned out to be quite a good model system based on the sequence and later on structure alignments, because the majority of the most important parts, they are very conserved. So especially transmembrane helix 8, which bears all the amino acids which make binding site for glutamate. So basically, whatever we see in archaea, to a certain extent, we can translate into mammalian transporters, and this is what we've been doing for many years. Obviously, nowadays, research has been shifted more towards the mammalian transporters. And why we actually need to study these transporters at all, I already mentioned you that this is one of the major neurotransmitters. And actually, if we have a look at the synaptic cleft, so we have it over here, so we have two neurons, and glutamate is released from the vesicles, and whenever it goes across the synaptic cleft, so it starts to stimulate certain receptors. So as soon as you have this signal transduction happen, you must immediately to pump out glutamate from the synaptic cleft back. Otherwise, you will have overstimulation of your receptors, and this is undesirable because in the end, it will actually lead to the death of those neurons. So you really have to keep this in great control. And those transporters, AATs, excitator amino acid transporters, the major role is actually to pump back glutamate. If we look at some pathologic conditions like ischemia, for example, so what happens, because you will have a reverse gradient of the membranes, these transporters will start working in reverse. They will actually pump even more glutamate into the synaptic cleft, causing the death of neurons. So this is a very undesirable effect. Hence, it's better to know the mechanistic details of this process. Another reason why it is important to study glutamate transporters, because glutamate is actually a very good to visualize brain tumors. So whenever you have brain tumors progressing, so you have a lot of glutamate, and this is possible to detect by different methods. And we will talk about later actually about some of these transporters which are overexpressed in cancer, because some of the members of this family, they are known to transport glutamine, and cancer cells, they really rely on glutamine a lot. So it's kind of very important in terms of pharmacology, in terms of physiology to study these transporters. And in terms of functional properties, they're very similar between archaea and humans, although there are certain important differences. So the main difference is that human transporters, obviously, as I said, they're involved in the regulation and transport of neurotransmitter glutamate. In archaea, they prefer aspartate over glutamate, and in archaea, aspartate is just a kind of food, it's a building block they consume. Hence, you can also already, without any scientific experiment, guess that in mammalians, these transporters work much faster, because as I already told you, you really need to do it quickly to pump out the glutamate. So in archaea, they don't really care. They just want to consume aspartate, hence they are slower. Another important difference is that, well, both systems actually fueled by gradient of sodium, so secondary active transporters. But in mammalian, you also have co-transport of protons and counter-transport of potassium. And in archaea, you don't find that. Also importantly, in both cases, there is uncoupled chloride conductance. And again, I will tell you later about some physiological implications of this phenomenon of having chloride conductance in these transporters. In terms of structure, all members of this family actually have the same general fold, and this fold is made of trimers. And in each protomer, you can see the scaffold domain. So here it is in yellow, and the transport domain depicted here in blue. So you have 3 transport domains linked or coupled to the scaffold domain. And I'll show you later that those transport domains, they work in so-called elevator fashion. So just a flashback. So for people in audience who as old as me or even older, so when we started to work on this topic, so state of the art was that cryo-electron microscopy was not even imaginable, because these proteins are too small. So people worked on something very big, right, like ribosomes or proteasomes, et cetera, So in terms of crystallization, crystallography, there was not yet like robotics. So we did everything by pipetting by hand. And crystallization screens were just a big market. So this is kind of very interesting for younger generations. So then I give a talk to students, they're kind of surprised, like, oh yeah, so 20 years ago you couldn't do this. Well, yes, we couldn't. But let's talk about function of this transporter. So I'll spend here some time so that you get acquainted with the way how we run assays with these transporters, because later in my slides, I will just refer quite often to, okay, we measured this, we did that, but how we do that. So what we do, so we purify the protein and then we reconstitute it into proteoliposome. So you have a synthetic mixture of lipids, or you call extract lipids. So you place your transporter, and now you have possibilities to vary the content inside of your liposome, proteoliposome, and outside buffer. And by changing those conditions, we can measure different things. So what we often do with these transporters, or what we always do with these transporters, we measure the transport of radioactive L-aspartate, because, well, it has a very strong signal, so we can easily quantify and see the level of transport. So, and this is what we did. So we have, let's say, the control is just the uptake of aspartate. So now if I add 400 times more of unlabeled amino acid to the radioactive L-aspartate, right? So what happens if I add to radioactive aspartate non-radioactive aspartate? I have no signal because it's so diluted, because I basically mix aspartate with aspartate, right? So the signal is gone. But if I add something which is not readily transported, let's say I had excess of L-glutamate, my signal is as strong as it was in the control. I mean, in this particular case, it's even larger, but this is just because the batch of protein was different. But you get an idea. So by using the excess of non-radioactive substrate, we can actually monitor how well this transporter can transport our substrate. And what we did as well, so we checked whether it is pH dependent and is it sodium dependent. So and then we replaced it with potassium. So it doesn't sense pH at all, so it doesn't really care, although there are certain members which actually pH dependent, but we are not going to talk about those today. Majority are pH independent, and it really depends on the sodium. So if I remove sodium from the system, I see zero transport. So it can't transport aspartate anymore at all. So we double check that also with ITC. So whenever you have sodium present, you can measure nicely. So you can see nice curves, and you have nanomolar binding of aspartate to the transporter. If I remove sodium, then basically I see no binding. And the question was, why? Why do you need the sodium to be present to have binding of aspartate? So we tried to answer this question, and here where structural biology comes handy. So what we did, we solved the structures of APO form of a transporter, so without any substrate present, and substrate loaded. And then we looked, well, here I have to introduce actually some elements again. So this is just the protomer now. So there's black, in black there is a substrate. One slide back. So this green one and orange one, those elements are helical hairpins. So they positioned like that, and they cover the binding site. And later I will show you that actually one of these elements works like a door. So it opens and closes, and that's the substrate in and out. And when we solved the structures and we did the comparison, so we noticed there is a particular region in the alpha helix, and this is unwound. So it's kind of very untypical, because normally you just have a helix, and well, it's a nice structure, right? Nice secondary structure element. But in this particular case, we saw that there is the methionine which kind of breaks this helix. And when you compare both structures, you can actually see that this methionine makes this movement, and this movement is quite significant. And this methionine is invariantly conserved in all the transporters, in the archaea and in human as well. So basically what we figured out, that you need to have sodium binding to shape the binding site for L-aspartate. So it's a bit hard to see over there on the right, but the whole idea is that when you have sodium bound, you trigger a cascade of conformational changes in the protein, and that cascade in the end actually shapes the binding site, high affinity binding site for aspartate. And the last sodium sort of binds and closes that door, and then the transport domain can move. So I think we'll move from here. The question was, is there any particular order in which binding occurs? And apparently there is. So we for sure knew already at that time that one of the sodium has to be lost. So the last sodium, before that aspartate, and before that sodium 103. So this, you can ignore 1, 3, 2 numbers, because it's just the way, well, we numbered those sodium sites. It doesn't really matter right now. And well, we'll go not into details here. So we developed kind of kinetic model, which in a way showed that it doesn't really matter which of the sodiums binds first. So what matters is that the very last sodium binds to the sodium binding on side 2. So we also developed handedness. So I told you about those proteoliposomes, but the issue there, you cannot really control if your protein is sitting like this or like that. So there's simply no physical way to control the orientation of your protein. Hence, actually, we developed C-bodies. So C-bodies is synthetic antibodies. So you have a ribosome display, et cetera, et cetera. And actually, we generated that, which particularly recognizes this part, this epitope of the transporter. So let's say if we add that C-body, we will inactivate all the transporters in this particular orientation. So this is also like a handy tool, how you can control the orientation of transporters. So we also did a lot of molecular dynamic simulations in an attempt to understand which sodium binds first. Again, I don't really have time to go into details here. It's kind of complex, but Markov modeling is a nice technique which actually allowed us to visualize the most probable trajectories of sodium when it enters from the bulk into the transporter. And it turned out to be the most probable routes, just a couple of them. So we more or less know which route sodium will take as soon as it enters the transporter. But let's go back to structures. So there have been myriads of different experiments, not only structural biology related, but let's say electromagnetic EPR, FRET, AFM, and all of them showed that this protein works in this elevator-like fashion. So you have transport domains, those in blue, which goes up and down, and they just bring the substrate from one side of the membrane to the opposite side. So here it's it's important for you to remember now that I will talk later about positions of those transport domains. So then they go up, so it's outward facing conformation, and then they go down, so this is inward facing conformation, and it will be somehow important during the lecture. And yeah, there are plenty of different conformations resolved. And the question was, can we get simultaneously transport domains in opposite orientations? Because all, let's say, structural biology attempts always showed that the domains are parked either outwards or inwards, but never, let's say, in such a position. And the question was, okay, is it just a limitation of structural biology, or is it an artifact even, or can they even do this? Is it stable enough in the membrane, et cetera, et cetera? And this is where single particle cryo-EM came handy. So this is the moment when our institute bought a microscope. At that time, it was kind of state of the art, I would call, not anymore. So now it's considered already old, and there is almost no technical support to some of the parts of this, like a camera. K2 camera is not supported anymore, for example. So I will not go into details of pipeline, so how we did, how many particles we took, et cetera, but I will rather show you kind of an interesting experiment, how we managed to get a different population of particles of transporters in different conformations. So we did kind of, well, I mean, some of it is obvious, so we just saw the structure without substrate. But this one I particularly like because what we did, we undersaturate the protein. So we did not give it enough substrate. And this is actually caused that some of transport domain are facing outwards and inwards and the other way around. So we really could, from one data collection, resolve the transporter in different conformations. And then we titrate it and saturate it, and then actually managed to put all the domains in a certain orientation. We also did an additional one with a known inhibitor, actually to push domains as far as possible outwards. And this inhibitor particularly prevents the closure of this HP2 door, so it cannot really move. So we resolved quite a lot of actually different conformations within this whole ensemble of structures we collected. I mean, again, I will not go into details here, but some of the states we observed for the first time. And one of such states was sodium-only state. So you have sodium bound, but you don't have substrate yet bound. And this is actually pretty cool because we could figure out how this transporter prevents spillage. And by spillage here, I mean that it would move without substrate being present. So you have only sodium, but let's say the substrate is not bound and the system wouldn't move. So that would be counterproductive. So you would just work for nothing, and nobody likes to work for nothing, right? So everybody wants reward. So it happens to be that whenever you have only sodium bound, this door of elevator, that HP2 element, cannot close. So the system physically cannot move. So you really need the presence of both sodium ions and aspartate. And only then actually the system closes and can move, and transporter works and does its function. So I think it's a pretty good result. So yeah, so we showed that this is actually this, HP2. This is the element which works as a door and opens on both sides of the membrane. So beforehand, people thought that perhaps, okay, HP2 works in the outward facing conformation and HP1 is very symmetrical, so it will open in the inward state, but it's not the case actually, because the movement is so large that actually HP2 can open on both sides of the membrane. And yeah, that's what I have basically told you already, that in the sodium only condition, it looks exactly the same as in the presence of inhibitor, so the door cannot close, so the system cannot go down. So what we also did, so because normally people do not like then you study proteins in detergent. The reason is because when you extract your protein from the membrane, the detergent, you may introduce a lot of artifacts. And well, this is the case in many studies. Hence, we did reconstitution into nanodiscs. So nanodisc specific scaffold proteins, which can actually embed your protein of interest with some lipids. So again, it's not ideal mimic of the real world, because the best would be to study it in situ, but it's certainly better than detergent. And we can actually show that the particular deformations of this nanodisc scaffold protein, which probably reflects deformations of membrane in the real world. So the interesting observation that we observed significantly smaller diameter of our nanodiscs. So in literature, it postulated to be about 15 nanometers. What we observed is about 10 to 11 nanometers. So our assumption was that what if it is an artifact of cryopreservation? So whenever you do cryo microscopy, so cryo, well, kind of obvious, right, that you need to freeze it. So you freeze it in the propane. So we thought maybe it actually shrinks, so when you freeze the protein. But then we did SACs with these nanodiscs, and actually in solution they showed the same size. So it's really not an artifact of cryo, but rather specific to a protein. So you have some flexibility in those nanodiscs. Can we improve the resolution further? Yeah, it turned out that, well, cryo-EM field is developing rapidly. So if you follow the field, you can see that every few months there is new software, improved software, a new protocol, something. And at some point, one of my PhD students revisited the data we collected in 2020, and he managed actually using some neural networks training, he managed to improve the resolution like almost to 0.4 angstrom. So that's kind of quite a lot in terms of information you can get. So the higher your resolution, the better level of information you can get from your data. So it does work for sure. Probably if we process it 5 years from now, we will get to maybe 2.5 or so. So it's also interesting that since it is reconstituted into nanodiscs, we could identify a lot of lipids, so those in magenta here, and they are sitting close to HP2 element. And HP2, I will just remind you, is that door of the elevator which opens and closes to let substrate in and out. So probably that movement is actually also lipid assisted. So you need those lipids there to make it faster. So I'll talk a little bit about our attempts with time resolved studies. So we always wanted to monitor the movements of that gate, HP2 gate or door. So initially we tried to do it with X-ray free electron lasers. We got the crystals, we got the time, but we never measured because, because of, well, COVID hit hard. So 2 years in a row, we couldn't do anything. And actually, interestingly enough, you can now do time resolved studies also with electron microscopy. So the scale is a bit different. So with XFEL, obviously, you can get the time resolution of femtoseconds. But to be honest, we don't really need that for this particular project because that movement is significantly slower. So in this respect, yeah, maybe it's a better choice actually because it offers you microsecond resolution. So to do this study, so actually, yeah, that's again the introduction of the elevator. I will skip this. We developed in collaboration with chemists photoswitchable compounds. And those photoswitchable compounds are designed on the scaffold of azobenzenes. And azobenzenes actually are pretty cool because, first of all, they are stable. Second of all, you can easily isomerize them. So you can go from trans to cis just by application of UV light. And importantly, you can actually also reverse it back. So if you shine the white light on it. So you can have this interconversion between two isoforms. And actually, it is pretty cool because, so again, if we just monitor the transport uptake of radioactive aspartate, and now we add this photo switchable inhibitors, so we can change the inhibition profile. So let's say if I start here with trans, I have very good inhibition. I switch it to cis, my inhibition becomes much worse, and then I can switch it again. Trans, the inhibition becomes better, and then I can change it to cis, and so on and so forth. I mean, obviously, at a certain point, the whole system will crash out. But nevertheless, that was a fantastic prerequisite for our time resolved studies, so we could see that indeed, well, there is a change in inhibition activity. So to understand better why it happens, we solved some structures, and we actually could show that indeed you have so the binding in the cis is not as good as in the trans because you have in trans additional hydrogen bond, which actually gives you higher affinity, obviously. So we also developed a cage compound, which you can actually cut with UV light, but I will move from on because it's still ongoing. So we're doing some EM studies now with colleagues in Lausanne. I will move now to a slightly different topic where we actually checked whether these transporters can actually also transport D-aspartate. Because it was debated in the field whether actually D-aspartate is even neurotransmitter or not, and even if yes, okay, which system will transport it. And it turns out to be that SLC is again a good good system for that. So what we did again the same study, like we checked the binding with ITCs. We did, well, the different conditions. We showed that the binding affinity is almost as good as with L isoform. So D binds slightly worse, and later we figured out why. And we, by plotting affinities versus sodium concentrations, taking the negative slope, we could identify the ratio that it's also 3:1. Yeah, these transport activities we will skip. This is a particularly interesting experiment where what we did to determine the ratio, we actually put different potentials on the membrane. And let's say if we have a certain potential which corresponds to certain ratio, so like in this case, it's 2:1. So what we had an influx of aspartate in our proteoliposome. So if we did 4:1 ratio, which is about 26 millivolts, so then we have efflux of aspartate, and only when we have 3:1, then we have net zero flux. So that basically means that indeed the ratio is 3:1. So again, not surprisingly, as the structural biologist, we solved the structure. We actually showed that both stereoisomers bind at very similar poses, although they're different by definition because you have different stereoisomers. And while there are differences in hydrogen bond lengths and angles at different ends, you have slightly different affinities. And we could show that the mammalian transporter will not fit the isomer of glutamate, because then you have a sterical problem, so you cannot really fit it. So you can fit in mammalians D in L aspartate easily. You can fit L glutamate, but D glutamate cannot be transported. So now again, I will switch a little bit when I will show you why chloride conductance actually matters in these transporters. There's a certain condition called episodic ataxia that people, well, have issues with coordination. Well, they can fall, they can have problems with muscles, headaches, etc., etc. And it turns out that different causes of episodic ataxias, and there are few which are well described, and they link to malfunctioning of channels. And surprisingly, EA6, episodic ataxia 6, was linked to a transporter. At some point, people were like, well, what does it have to do with transporter of glutamate? And well, it was actually even pinpointed down to a particular mutation of proline to arginine in a certain transmembrane helix. And well, we asked ourselves why. And at more or less the same time, other groups showed that indeed there is this formation of chloride channel in these transporters. And the whole idea in the field was, so this is this particular position in one of the transmembrane helices. And as you might know, whenever you have proline in the helix, it just breaks it. So it makes a kink. And the whole idea in the field was, okay, if this particular mutation of proline to arginine, most likely case scenario is that you release the kink so that you will actually straighten up the helix. And that would have implications to the transport activity, blah, 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 blah. So we decided to verify whether it is the right thing or not. So we produced the mutant. Again, I mean, the assay did as I explained to you. So we checked the transport of radioactive substrate. We did different ratios of protein to lipid. The system obviously works. So we have a robust uptake. We did, again, ITC, so we have very good affinity, good binding, but this is interesting. So when we compare wild type to this particular mutant, so we actually have a drop in Km, so in transport activity. So we have reduced transport, which is obvious from Km values, but the affinity to substrate has not been changed, which is kind of a little bit puzzling to us. It was at that moment. So what we did next, we did so-called surface supported membrane electrophysiology. So this is an interesting setup. I mean, again, I don't have a lot of time now to go into details, but very briefly what you have, you can, again, just have your proteoliposomes, you put them on a certain electrodes, and you can measure currents. So it is much faster and much cheaper to do a classical electrophysiology and actually works pretty well. So we use double exchange protocol, which means, I mean, again, I don't want to go into technical details, but what you do, so you have non-activating solution when nothing works, then you have activating solution and systems start to work. Then you have, again, non-activating and resting solutions. So you basically just change the solution. So depending on what you want to measure, you come up with your buffer composition. So in this particular cases, we wanted to measure two things. We wanted to measure currents related to sodium transport, and we wanted to measure transport related to chloride transport because we're looking into chloride conductance. Although we substituted chloride with cyanide because cyanide has much better permeability and much nicer signal. And importantly, what we saw, so when we have a mutant, we have much higher anion conductance. So it really, really goes up. And so we have reduced transport, but increased anion conductance. So then it was a time to solve the structure. Again, we used cryo-electron microscopy. We solved it. And to our surprise, although we obviously see that this is a mutant form because we see arginine in place of proline, the kink is still there. So the helix maintains its integrity despite this is kind of very substantial mutation. So the idea that it will straighten up the helix didn't hold. But what we noticed that at this particular position, this arginine is actually facing lipid bilayer. Again, not a very typical situation, but apparently this arginine is docked to one of the lipids, hence it will, again, I will not go into details of the dissimulations, it will hold this transport domain and preventing it from movement as efficient as it should. So hence we have, well, indeed reduced transport, but increased chloride conductance. And that's the reason why this causes this episodic ataxia 6. So I don't have that much time, but I still want to tell you about neutral amino acid exchangers. So two of them, ACT2 and ACT1. So ACT2 was shown by many to be linked to many cancers. So it's really overexpressed in cancer. And it turns out that ACT2 is very agile in transporting glutamine, and cancer cells rely on glutamine uptake. And again, we did exactly the same kind of experiments, same transport of radioactive assays, and we saw that this is very good in transporting glutamine, but also alanine, serine, cysteine, threonine, and this is actually from their original names comes ACT. But also, well, it can transport asparagine, but no bulky and no negatively charged amino acids. So we solved again cryo-EM structure. I will not go into details. What is particularly interesting, it looks very much like all the structures before, but now we have so-called antennae here. So these protrusions, and those protrusions are actually recognized by retroviruses. So this transporter also serves as a receptor, which on one hand, well, helps something like fusion of cells during placenta formation, but on the other hand, is exploited by many viruses for the entry into the cell, which is very unfortunate. So there's very similar binding between AATs and ACT2. There is just one important change of arginine, which is replaced with the cysteine. And well, there are also extra densities which are assigned to lipids, etc., etc. We'll skip this movie, but we'll get back to the synaptic cleft now, because I want to last piece of my talk to talk a little bit about ACT1 transporter. So it's a very complex system. So again, you have fluxes of those neurotransmitters. You have different receptors. Accepting the signal, you have different transporters pumping, well, certain ions back, pumping in and pumping it out. And now we'll focus on CT1. So it's very similar to ACT2, but their profiles are very different. So importantly, ACT1 is really overexpressed in the brain of both healthy people, but also people developing cancers. And the question, why is ACT1 is so much expressed in the brain? So again, from the name, so you see it's alanine serine cysteine transporter 1. Turns out to be that it's very important for the transport of L-serine. So and why you need L-serine? Because it's a precursor for L-cysteine and for D-serine. And L-cysteine you need for production of glutathione, and D-serine is important for NMDR receptors. Turns out also that there is a very significant production of D-serine in neurons, but L-serine is produced in astrocytes. So you need some sort of serine shuttle between astrocyte and neuron. And the main role of transport of L-serine is ascribed to ACT1. And again, we showed with radioactive transport assays that this indeed very effective in the transport of L and actually D-serine, but it cannot transport glutamate. So why it is important? Because mutations in ACT1, they lead to terrible conditions. And those conditions in general called SPATCCM disease, and that means spastic tetraplegia. Then it's almost like paralysis, so then people cannot control their muscles. Thin corpus callosum, so then the connection between two hemispheres of brain is very weak, so they don't really interact with each other. Progressive microcephaly, so then the brain is not really growing as it should. And you can imagine that this is really terrible. So the development delays, inability to speak, inability to walk, intellectual disability, and so on and so forth. And importantly, these conditions develop even if ACT2 is intact, despite it was shown that ACT2 can also transport L and D-serine, it doesn't really help. So again, we solve the structure. We'll not stop here. Will not stop here. It looks almost exactly the same. Binding site, again, exactly the same, but now instead of cysteine, you have threonine as the main binder. And there are several mutations characterized. So few nonsense mutations, so tyrosine 191. So you can imagine you produce just part of scaffold, but no transport domain. There is tryptophan 453. You produce almost complete protein, but you miss part of a transport domain, which actually binds the substrate, and you have frame shift. So you can imagine that that protein is useless. And the number of characterized missense mutations, and we looked up at those. So we produced E2562 lysine mutation. So we measured the transport. It can actually transport to some extent the substrate, but what happens, and we did MD on that, so it docks the transport domain, so it cannot move as efficient as it should. So we analyzed arginine to tryptophan, and this is really killing mutation, so you can't really transport anything. And what happens, it actually pulls HP gate and doesn't allow it to close or open. And the last mutation is very destabilizing, so it actually just breaks the whole site. So we could not even express that mutant. So I think here I will actually stop. So I will not go into conclusions. There are many conclusions. There are many outstanding questions. There's been plenty of people working. There are probably several generations of PhD students. So I'll be happy to stop here to answer your questions if you have any. Thank you.
Thank you very much. Great talk. Many years ago, it was shown by an Italian group, by David Trotti, that these proteins E82 and E81 were very prone to oxidation. And when the transporters were oxidized, they actually pumped glutamate out from the astrocytes, increasing the level of extracellular glutamate. And that was a hypothesis that was considered to be a root cause for neurological diseases like amyotrophic lateral sclerosis. Have you ever worked with such kind of modifications to the structural composition of the proteins under oxidative stress or something like that?
Yeah, thanks a lot for this question. So we haven't looked really into that, but you're absolutely right. So basically what happens, you can switch the way these transporters work because they don't really care about which way to transport. I mean, the way from this side to that side and the reverse is the same. So they do not sense. So it really depends on the condition you maintain within the cell. So if you change the conditions, you just change the direction, which is unfortunate actually for many cases.
Thank you for your talk. I have a question related to the first part of your talk. When glutamate and sodium are transported to the other side of the membrane, is also sodium released first and then glutamate? I mean, do they follow the same kinetic of transport?
Yeah, so from kinetic model, it seems that again, so there's certain things like first one of the sodiums which is closing the door has to be released, otherwise you will never be able to open the door. Then most likely substrate, but there is a debate. So actually by our kinetic model, it seems like there are several possibilities which are nearly equally possible, and it doesn't really matter. So what really matters, the order for the binding, so you need to have at least one or better two sodiums bound first, then you shape the side. Otherwise, the system will not work.
Thank you.
Do you have any idea of how this
Yeah, so I was probably too quick there. So the whole idea was that there are two possibilities. So is it diminished transport or increased anion conductance? So it seems like the main reason is increased anion conductance to actually deplete your cell of chloride, for example. So it shrinks in the end. So, and well, dies out and doesn't perform that well. So it's not as severe as, let's say, this spot CCM disease. So many people may not be even aware that they have this episodic ataxia. It can be extremely mild. But, well, in certain conditions, it can be more severe. But I think the consensus now that the main reason is increased chloride conductance. And now we basically showed that it happens because this arginine is actually docks the transport domain to the lipid bilayer. So let's say it doesn't go that efficient, so it really stays there. And the chloride conducting channel is formed while this transport domain is moving. So let's say if I have situation like that, there is no chloride conductance. I have situation like that, there is no chloride conductance. I have situation when it's moving, there is a formation of this channel. So it's transient in a way, and arginine stabilizes it.
So this channel where the transport modules, at some point, they all start going up and down at the same time. But first one starts, and when you increase the sodium concentration, all the other transport modules start to move.
So they move independently from each other. So
independently.
Yeah, there's been series of experiments. I mean, like Fred, I think, really gave an answer. no cross communication. So they don't really know what the other domain is doing. So they don't even care. So the question then, why do you even need a trimer? So the explanation is that most likely, first of all, it makes it more stable because this is kind of large and disruptive for membrane movement. But second, so you have this bowl-like shape. So you have increasing local concentration of your substrate. That makes it efficient system.
Thanks.
Well, if there are no more questions, thank you so much, Albert, for this great talk.
Thank you.